Hi, I'm Julia.
And I'm Jordan, and we have been doing a research project on phytoplankton concentration going in and out of Biddeford Pool. Our project is titled Phytoplankton Concentration in the Spring and Fall High and Low Tides.
Abstract for our study is that preliminary data is being gathered on the concentration of phytoplankton flowing into and out of Biddeford Pool. With the knowledge of the pattern of phytoplankton concentration during flood and ebb tides flowing into and out of Biddeford Pool, it can lead to further questions of what happens to the phytoplankton during their time in Biddeford Pool. For example, do they become prey to other organisms or do they settle at the bottom? Samples were taken 4 times a month during the spring and neap tides. Water samples are collected in the mason jar on each day at 2 different locations, the bridge and the mouth, for flood tide as well as ebb tide. Data was collected during spring and neap tides. Spring tides occur during full and new moons, while neap tides occur during the first and third quarter phases. Samples are filtered through a 10 micron sized net and run through the flow cam where phytoplankton is identified and concentration is calculated. It is hypothesized that there will be more phytoplankton found in the flood tide at both sampling locations, but the total cell count will be lower than the summer because of seasonal shift. The results show that at both sampling locations, there are more concentrations above zero, meaning that there is phytoplankton entering the sample location than leaving. Overall, the concentration of phytoplankton was higher at the bridge of Biddeford Pool than at the mouth. In other words, more phytoplankton is entering Biddeford Pool than exiting.
Water samples were collected in mason jars at the bridge and mouth of Biddeford Pool by scooping water into the mason jar, filling it at least halfway. In the lab, each water sample is filtered through a 10 micron net to separate all of the collected debris from the phytoplankton. Then the filtered water from each location is separated into 3 50 milliliter Falcon tubes. Volumes of the initial water samples in the mason jars, volumes after filtration, and volume of each Falcon tube is recorded. In total, there are 12 Falcon tubes of 50 milliliters that are run through the flow cam, 3 for each sample location for both tides a day. Each sample runs through the flow cam for 20 minutes with a 0.15 milliliter flow rate. Google was added to the samples that were not finished on the same day.
The data shows that there is an uptake of phytoplankton concentration at the bridge sampling location. After taking the difference between both tides at each location, the concentration for each location are greater than zero, meaning that there are phytoplankton species coming in but not leaving. Species that showed up fairly often were Skeletonema, Coscinodiscus, Gennardia delictula, Catoceras, and Alexandrium caturnilla. Skeletema had the most richness in concentration with 889 individuals that were captured on the flow cam. We were able to capture 406 individuals in the genus Coscinodiscus, 272 individuals from Grenadier, 137 from Catasaurus, and 130 from Alexandrium.
Our results prove that a larger concentration of phytoplankton are coming into Biddeford Pool compared to those leaving. This can correlate to phytoplankton being eaten by predators such as zooplankton and slowly making their way up the food chain. Most of the species found are highly toxic species of phytoplankton and do contribute to harmful algae blooms. Harmful algae blooms lower the water's oxygen levels and can cause massive fish kills in areas where oxygen is completely depleted. They are caused by excess nutrients in the water, causing toxic phytoplankton to produce toxins that can impair functions of fish, shellfish, seabirds, and even mammals. This is a preliminary study, and we are still trying to figure out the most effective way to collect and analyze data.